is because the mature follicle is actually hard to define. It's hard to really tell the difference. Is it a big secondary follicle or is it a mature follicle? If I were a mature follicle, I'd actually like to see it near the germinal epithelium. And so our textbook and our histo book actually used to contradict. Interestingly, they showed the exact same picture. One of the books called it a secondary follicle, the other called it a mature follicle, just showing that it's hard to differentiate. So I don't want to do that. So I actually will say you don't need to worry about seeing the mature follicle in the microscope. I think it is hard to define and be absolute with that. However, you do need to be very comfortable with a high-power view of the secondary follicle. So this is actually one thing that you can see looks an awful lot like the slide we were looking at. The entire structure is a secondary follicle. And just to make sure you have this is a secondary follicle. What cell is that? It is a primary oocyte. Remember, the primary oocyte does not become secondary oocyte until the mature follicle. Very, very common mistake. And so, but it is still a primary oocyte, even though we're looking at a secondary follicle. The cells there are the granulosa cells. Good. More than one layer. Granulosa cells, they're doing secreting estrogen, inhibin, and follicular fluid. And these cells now, notice these are granulosa cells, but they're special granulosa cells, lucky granulosa cells, that are surrounding the primary oocyte. These particular granulosa cells form a cellular layer called the corona radiata. And so I have that in that picture. You want to be able to view it here. The corona radiata is actually going to be ovulated. This is the mature follicle that we will not be looking at in the microscope. You do need to know mature follicle in the models, but not in the microscope. And so let me move on to, just to also emphasize, if you have this model, and when you're looking at this model, probably the best thing to do when thinking about the oocyte is these are all primary oocytes. Then it's in the mature follicle, this one, where that primary oocyte becomes secondary oocyte. Therefore, this little white thing right there, I wouldn't ask what is the cell, because you would know it could be primary or it could be secondary. This is where it changes. And that's helpful when you're looking at this model. You say mature follicle, this is where primary becomes secondary oocyte. And so that is where, what hormone causes meiosis to resume? Do you remember from last year? It was LH. The LH surge causes meiosis to resume and causes the ovulation. And so here you can see the ovulation of that mature follicle, and therefore this cell is a what? Secondary oocyte, for sure, because we have ovulation right there. And so that is a secondary oocyte. This is what is ovulated. And so just to remind you, this white thing here is the thing that is ovulated. The cell there is the secondary oocyte. So this is secondary oocyte. So it became secondary oocyte, mature follicle. Now you have the ovulated structure. So that is the secondary oocyte. You also, remember, have the polar body, first polar body right there. And then you also have some protective layers. This is something we'll see in the microscope. This is an acellular layer, meaning there's no cells there. But there is a kind of hard gel-like substance, and this is called the zona pellucida. It's a protective barrier for that secondary oocyte. I mentioned in lecture that sperm can break through it. Only human sperm can break through a human zona pellucida. And I put here that it is an acellular layer made up of glycoproteins. One example is hyaluronic acid. Oops, hyaluronic acid, the enzyme that breaks it down, is hyaluronidase. So hyaluronic acid, good one to know because it is the main chemical in connective tissue. It's what makes cartilage hard. It's what makes, it's what makes cartilage a gel-like substance as well, or just connective tissue a gel-like substance. And so this chemical is in the zona pellucida. It gives you an appreciation for the zona pellucida. It can become pretty hard because it is has these glycoproteins. And so it has hyaluronic acid. Good chemical to know. This is also the one that makes the joints, it's in the eye, so when you feel your jelly eyes because of hyaluronic acid. Hyaluronic acid is also what makes the synovial fluid very lubricating, but it also is what makes the cartilage very hard. So good chemical to know. There is hyaluronic acid and some other glycoproteins in this zona pellucida. it is protecting that secondary oocyte. And so an important layer right there. And then there's the cellular layer, which is the corona radiata. I mentioned this already in the microscope. You see the development of the corona radiata. And so the cellular layer right there is the corona radiata. And so remember, we are seeing the development of the corona radiata right here. And so you can see the oocyte there, and this is corona radiata, and then those cells there will be ovulated with the oocyte. And so we can see corona radiata in 2 places right here. And so you could be at either place. And so now when we look in the microscope, we just did this one, identify this cellular layer right there. Corona radiata. And so now we can see it in the microscope. This is all corona radiata. If we go to high power, what we'll do is just go to high power like this. This is the picture I drew right here. And so you can see corona radiata right here. There is a layer right here. This is the primary oocyte. So this layer is zona pellucida. And so you have zona pellucida right here. It looks clear, but there is a, hyaluronic acid, some gel-like substance right here. And then you have the antrum. You can see where we're at right there. So you have the antrum with follicular fluid. The cells right here are granulosa cells. One thing I like about this view right here, notice granulosa cells right here. A little antrum is being formed. You get the idea that these granulosa cells are secreting out that follicular fluid, and the follicular fluid just starts to collect up, and it forms a little antrum. And so eventually these antrums will all kind of accumulate into one big antrum. But I've drawn my picture here. You want to be comfortable with the zona pellucida, corona radiata, and you have granulosa cells. Still primary oocyte. And so it's still positive in prophase 1. Questions?